Well, as everyone comes back and gets settled, it's always a good sign when it's hard to get people back from a break, because everyone likes to be together and to talk. So it's great. It's great to see that. And it's great to have some conversations from the present and from the past. You will recognize that I am neither Chris Prather nor Paula Hammond, but I have the privilege and pleasure of introducing Chris. Chris is the Arthur D. Little Professor of Chemical Engineering at MIT. She did her undergraduate in the department before going off to Berkeley to get her PhD, where she had worked for Jay Kiesling, many of you know. And she then went to Merck for a number of years and experienced the real world of process technology and biochemical engineering. She joined the MIT faculty in 2004, and her research is focused on novel bioprocesses that are based upon recombinant microorganisms. And I've had the good benefit of sitting on thesis committees of a number of her students. Her honors include being named a fellow of the Radcliffe Institute for Advanced Study, as well as the American Association for the Advancement of Science. In a fitting continuation of Dan's legacy, she has been honored as an outstanding teacher, receiving the C. Michael Moore Outstanding Faculty Award, one that is given with great appreciation by the department for excellence in teaching, following our distinguished colleague, Mike Moore. And also and she's also a McVicker Faculty Fellowship, another very distinguished award at MIT. Chris is an entrepreneur, co-founder of Calion, that is using real bioprocesses in the real world to make real products. Please join me in welcoming Chris for her comments to the podium. Please, Chris.
So good morning again, everyone. I actually cheated. I left my glasses up here so that I can see them. And I made some notes, which some of you know that I like to wing it, meaning I like to just put some slides together and then talk about whatever comes to mind. And I'll do a little bit of that. But there are some important things I wanted to make sure that I would say. And so you'll see me referring to some notes from time to time as well. I want to start by talking about how I first got to know Professor Wang, as he was first known to me. As Charlie mentioned, I was an undergraduate here at MIT. It turns out I came at the same time that Arba Lamazé-Smith did. I arrived in the summer of 1990, fresh out of high school in Northeast Texas, ready to start my undergraduate career. I was pretty sure I wanted to do chemical engineering. I was pretty sure I was interested in biochemical engineering. I was pretty sure I had no idea what either of those really meant, but they sounded pretty good, and this was the early 90s, and so this was that sweet spot of actually seeing what biotech could bring in terms of real innovation that Phil and Charlie talked about earlier. So I arrived at MIT right at that time. I also thought material science was kind of interesting, and so I dabbled in that as well. And then in the spring of 1993, Professor Charlie Cooney was my instructor in 1037 kinetics. And I did pretty well, and I knew I was interested in graduate school, and I thought I should probably go ask this guy for a letter of recommendation. And then I knew that there was this thing called UROP, and so I went to see him and said, can I do a UROP in your lab next year? And he said, yes. And I said, can you tell me where I can find biochemical engineers to go to graduate school? And he said, yes. And then he said something that I remember he says, I don't know if he remembers, he said, and I would be happy to write you a letter of recommendation. And so I started UROP in the fall of 1993 with Jack Pryor, having no idea still what biochemical engineering was, and really didn't get my first introduction to what it meant in a formal sense until the spring of 1994. So my senior year, I've already applied to graduate school now, so I'm not that concerned about classes anymore. I was finally able to learn some stuff. Jack was very patient with me because in the fall, he was trying to teach me a bunch of stuff, and I had no idea what he was talking about. He was speaking completely foreign language. And then I actually took this class where I started to get a lot of the formalisms around biochemical engineering. You can see the class was taught by Charlie and by Prof. Wang, and it was hard. Not gonna lie, we were at dinner last night, and Doug Cameron talked about getting a low score on an exam that was because of a mistake. I got a low score on an exam that was because of a lack of knowledge, right? There were no mistakes associated with it. And I found this picture from my yearbook, my senior year at MIT, and I found this picture from the department's research guide my senior year as well. So I imagine that this is about what Prof. Wang looked like at that time. This is not what I looked like because I went to school with my hair pulled back and barely with eyes awake. And I remember him being very scary, right? Very intense, the intensity that we've all talked about, but also clearly interested in communicating knowledge, not just information, but actually communicating knowledge, getting a deeper understanding of what he was trying to teach, and not just really going through the textbooks. You can actually see that the required text was copies of biochemical engineering literature, which is not the name of a book, right? And the manual for Biopro designer, right? So this was a class that, by the way, I'm an undergraduate. Most of you guys took it as graduate students. There was one other undergrad in the class. He and I banded together in our misery around misunderstanding and misinformation and lack of knowledge. But coming out of the end of it, we really felt empowered. We really felt that we were part of something that was really making a difference. And so as Charlie mentioned, I went to Berkeley for graduate school. Matt Krogan showed a picture of his family yesterday, his Berkeley family. Those are my people too. My uncles, Uncle Harvey, as we lovingly call him. We didn't call Doug Uncle Doug, but Harvey was Uncle Harvey. Doug was chair of my thesis committee. I worked with him very closely, and I learned about working with microbes with Jay Kiesling, as Charlie mentioned. I decided to go to Merck, but not before I had a trip back to MIT. So in the 1998 AICHE meeting, I met the two Bobs, Bob Brown and Barb Armstrong, who said, oh, we hear you're doing well at Berkeley, and maybe you'd be interested in an academic position. I did not tell them then I had no interest in an academic position at MIT. That's a story for a little bit later. But they said we should keep in touch, and then I got an email from Prof. Wang that says, why don't you come out and visit during IAP in 1999? And I still remembered Prof. Wang is scary, right? But I also remembered something that Nubar said last night, which was it was very clear that Prof. Wang also rocked, right? And so when I first came in January of '99, he introduced me as one of his former advisees. He said, oh, you're my advisee. And I said, no, Professor Wang, I was not your advisee, but I did take 1059 from you. And he says, yes, right, okay. We had a wonderful conversation. I said I was interested in an academic career, but I really wanted to go to Merck first. I went to Merck. I met Dave Robinson, who was my department head. I first got to know John Onens there. Really had a wonderful experience in my time at Merck. And while I was at Merck, decided I was interested in coming back to academia. Still was not interested in MIT, but figured why not, I would give it a try. And Danny was a very gracious host even at that time. And when I interviewed and accepted the position, I had a year before I was coming back to MIT. And he said to me, this microbe stuff is going nowhere. You need to learn how to do mammalian cell technology because that is the future. And so I went back to Merck, and our department had been more microbial, was moving into therapeutic protein production. And I said, I actually asked, they said, we're going into the mammalian cell world. And I said, I'd like to learn how to do mammalian cell stuff. Danny Wang tells me the microbial stuff is going nowhere, and I want to see what the mammalian cell stuff is all about. So I came back to MIT in 2004, as Charlie mentioned. I decided, I'm sorry, fellow scientists and engineers in the audience, that mammalian cell culture was not my thing, and I thought microbes still had a future. And when I came back, Danny had some assignments for me, not surprisingly. And the first assignment he had is, you're going to teach my class. All right? He didn't say, would you like to teach my class? He said, you're going to teach my class. It hasn't been taught in a while. It's no longer 1059. It's 10542, 10442. This class is really important. It is the foundation of the biomanufacturing industry that we have today, and we can't lose the knowledge, and so you're going to teach my class. And so I actually said, okay. I thought this was great because I still had my notes, right? Like this picture was from my actual Bible on my shelf in my office, and I thought, great, I have the notes. I'm going to follow mostly along with what Danny did. I did make some changes to the curriculum. I did not tell him at the time that I made changes to the curriculum, but I made some changes, introduced some more molecular aspects around cellular engineering into the curriculum, and said, well, you know, it seemed to work pretty well. I'm going to follow along the same process. We're going to have 4 quizzes. We're going to have problem sets. We're going to have class participation. I made a fatal error in judgment. I did not realize that in addition to Charlie and Danny, Margaret was a TA. I did not have a TA. And it turns out if you don't have a TA and you give 4 quizzes, you have to write 4 quizzes, and you have to grade 4 quizzes. So the second time I taught this class, it had 2 quizzes and not 4. But there was a lot of enthusiasm. BPEC was gone by then, but there was still a lot of enthusiasm for this work and for this technology. And I really took a lot of what I learned in taking 1059, as well as the time I spent at Merck, and tried to figure out, how do I teach in the way that I remember Prof. Wang teaching, which was not about just delivering knowledge and information, but making sure that people really got it. They really understood it and they could put it into context. And so I unfortunately have not been able to teach this class in a while based on other commitments I've had administratively and other teaching responsibilities. But I consider this to sort of be the first flame, and I'll describe what I mean by the flames at the end. But this was sort of the first flame in terms of Danny saying, here's what I think you should be doing. The second flame was a different class. This class was a new class, a relatively new class called 1028. And I actually pulled this from some papers that I found in Danny's office. Danny had lots of paper that proposed creating a new course that would be a companion to a new curriculum that had been created, which was a new program in chemical and biological engineering. And what struck me as I read through some of the papers was that it was clear what Danny was trying to provide was not just a curriculum, but an experience. He gave a lot of thought to not just what should go in this class, but what students should be learning and what they should be learning in the context of what was happening in the industry much broader. And he worked very hard to put a class together that was proposed to be offered in the spring of 2003. Anyone who's been in academia knows that things that you propose usually take a little bit longer to actually implement, but he did get the class approved, and in the fall of 2003 offered for the first time a course in biological engineering, which was really and continues to be a bioprocess engineering course. So it was really the first time that a group of undergraduates could come into a lab at MIT and learn to work with the equipment, both the fermentation equipment and the analytical equipment that accompanied it, and learn these valuable skills that we learn were actually quite prized in the industry. It's still somewhat rare to have undergraduates who are trained in the classroom and using this equipment that's so fundamental to the industries that Phil talked about earlier today and that Charlie talked about as well. It was a massive undertaking, so this is the teaching staff for the class, the first time that it was taught, and you can see all the different individuals that are involved there. And so fall of 2003 was the first time the course was taught. Fall of 2006 was the first time that I taught the class, and I taught it with Danny and also with Jean-François Hamel. And I remember very clearly sitting in Danny's office with Jean-François a couple of months before the class was supposed to be offered, and Danny said, "This class is perfect. Do not change a thing." And he also said, "And we're going to do this together one time, maybe 2, and then it's going to be yours." And he went on to say, "You know I'm an institute professor. I don't have to teach. I teach because I want to and not because I need to." Okay? And that was an important lesson as well, which is that the best teachers are the teachers who teach because they want to and not because they need to. So I came into the class in 2006. I did not change anything the first year. It was a couple of years later when Danny stopped teaching. I did not change everything, right? But I changed many things, including the fact that we just really couldn't sustain a teaching staff of this size indefinitely. But the class continues today, even though I'm not primarily responsible for it. It continues to be well received by the students, but it also continues to be well received by the employers who take those students and are able to benefit from their knowledge and their experience. And I want to say again, the goal here in creating this course, and it's the spirit that still lives on, was not in putting together a curriculum, but actually in putting together an experience that would be worthwhile. So these were the classes that I inherited from Danny that were taught here at MIT. This is the second flame. The third flame, I'm going to touch upon a theme that actually came out yesterday as well, which was that Danny was not content to have influence on this continent. He really believed very strongly in the value of what we were doing as a discipline and the value of the industry that was being created. And he felt very strongly that that needed to be exported and developed in other parts of the world. And John Onens yesterday talked about the country of Portugal, and one person in particular, Manuel Carrando, who really worked closely with Danny to try to revitalize, and not to try, but to succeed in revitalizing the biotech industry and transforming it in the country of Portugal. Some of that work was done under the auspices of the MIT Portugal program. Charlie was a big part of this program as well, focused on entrepreneurship. And one of the initial themes in that program was bioengineering systems. It included biomanufacturing in the way that we've been talking about yesterday and today, but also included many other aspects of bioengineering as well. And this was a PhD program, the goal being to bring students from 4 different Portuguese universities into a cohort program where they would first have some fundamental classroom knowledge and learning. They would then do thesis research in Portugal, but many of them had thesis co-supervisors here at MIT. I ended up with 5 such students who then were able to come to MIT and do a part of their work there as well. And the way the program was set up, the courses happened in modules, these 3-week intensive modules. And Danny actually started the module in bioprocess engineering. Now, as legend has it, I don't have proof of this, as legend has it, the goal was to always have co-teaching between MIT faculty and Portuguese faculty. Legend is that the first time it was taught, Danny said, I'm doing it because I know how it should be done, and I'm going to do it this way so you can watch me do it, and then you can do it that way next time. Okay? I don't know if that's true, but that's the story I heard. Okay? And then after a few years, it really was this relationship between MIT faculty and faculty in Portugal. And I got involved in the class in the fall of 2011 after Danny became ill and went to Portugal and spent several days there. And by the end was spending close to a full week teaching in the course. Now, what was very interesting to me about this particular module within the bioengineering systems program is that part of what we were supposed to be doing in Portugal was to help build capacity. So co-teaching was supposed to be a transitional period into solo teaching by Portuguese faculty. And when the time for that transition came through, this class was the only class that was really ready to stand up and be delivered independently. And in large part, it was because of the relationships that Danny had built that included folks like Joachim Cabral, who had spent time here with Charlie and with Danny, and Joao Crespo, who had spent time here with Danny and Charlie as well. And so this course does live on. It's being presented in the same way, some tweaks I imagine, but in the same spirit that it was originally created for, because of the commitment to long-term capacity building that Danny had, and not just a fly-in, fly-out sort of approach towards international relationships. I do want to talk for a minute about Manuel. I first met Manuel actually through John Onens, right before I was supposed to be coming to MIT, where John had Manuel convinced that I was coming to MIT temporarily on the Merck academic leave program. I'm not actually sure how long Manuel believed that. Maybe John can tell me when he finally told him that I wouldn't be coming back to Merck. But I made a connection with Manuel there, and as John mentioned yesterday, he wasn't able to be here today. And John summarized some remarks that he had, but I thought what he had to write was so poignant that I wanted to be able to share it in his own words. And I'm actually going to read it here. There's about 300 folks or so who are watching live stream. I think they're going to have a hard time reading that text. So I'll take a moment here and say this. This is from Manuel. All I want to do here is to give note of how phenomenal Danny was in support to myself, and inviting me to join BPEC for 9 months in 1988, and later for IBET, of which he was a scientific advisory board member for almost 20 years. This meant he would travel to Lisboa, using 2 to 3 days per year to give advice, which was very relevant and forward-looking, at a time when we were just a concept, and IBET could only pay an economy ticket and a 4-star hotel nearby. A few times, Vicky would join Danny, becoming a friend to the Carandos. It was Danny at an SAB meeting that made the point in front of the plans for the building: if you do not build a BSL-2 laboratory now, you will never do it later. We built it, and now it is the core of IBET's molecular vectorology for gene therapy and viral vaccines. He will be critical, as you well know, with our ideas for projects or areas of research. Manuel, you've got nothing. If you do not get $5 million, you will go nowhere. But then always would go after ways of supporting us with contacts and introductions. Danny opened his lab to other IBET researchers, in particular the current CEO, Professor Paula Alves, and supported in many other ways in his mentoring of IBET and ITQB, our academic partner institute, leading to the Universidade Nova de Lisboa, bestowing upon him an honorary PhD degree. I will keep his presence alive at IBET and wish that the ceremonies will be up to what his strong commitment to MIT and his students deserve. So again, I wanted to read that just to reflect in his own words the fact that Danny's work was transformational across the globe. The last thing that I want to talk about also speaks to this transformation well beyond MIT's campus, and this is the fourth flame, and it's the summer course that you've heard about. So in the summer of 2006, Danny invited me to teach in this course for the first time, telling me again, one day you will own this course. You need to understand how to run it, and you need to be ready to step in when I'm ready to hand it off to you. In 2006, when I first taught, I took one of Danny's lectures, his lecture on modeling and monitoring of bioprocesses, and then introduced, slightly over his objections, a couple of lectures that had a little bit more of a focus on molecular biology and microbial systems, because I really thought this was an important place that the future was going. I should point out that what Danny would always say is that fermentation technology was a name that never changed, but the content did. And so this was a course where he did not actually feel that it should be static. He really wanted us to be improving it over and over again. And so here was a place where change was not only acceptable, it was required for us to continue to innovate and provide for anyone who was interested the ability to be able to learn more about these fundamental processes that were really changing the world. So I worked with Danny together on this course for several years, becoming in 2011 an official co-director of the class. And there was one change we made that first year, and that was in the snacks. We continued to have ice cream, and we continued to have cheesecake, and we continued to have cookies, but we added grapes and bananas and oranges. And one day, Danny walks in with a plate of cheesecake and says, "This stuff is terrible for you." I said, "Danny, I can get you grapes." "I don't want that crap." So as Vicky said last night, he was very much committed to his sweets, and we were very much committed to providing them for him. So I do want to spend a little bit of time talking about the course. This was the first slide Danny gave me to say, here's how the course is doing. I love this because you can see year after year after year, he got his tracing paper. If he made a mistake, he would correct it. And he plotted out the chart of the course. But then he did find Excel, and he put it all there. And by the way, I do not chart attendance from November. I decided this was the lag phase I was not interested in. And so sometime around late February is when I start tracking enrollments. And what I thought was always interesting is Danny would say the course was a leading indicator of the economic state of the country, okay? And if you see, when was the last Great Recession? Pre-COVID, when was the last Great Recession? 2008. Look what happens in 2007. right? Riding high, the course numbers drop in 2007, there's 2008 and 2009. So after this year, Danny said, uh-oh, something is coming. Nubar, I don't know if he spoke to you about ways to rebalance his investment portfolio, but he was convinced by that one metric that there was something coming on the horizon. So the class has persisted for a long period of time. And a few years ago, we got to do something really, really fun. We had a big celebration for the 50th anniversary of the course. Now, I will point out to you, we were celebrating the 50th anniversary in 2014, and it started in 1962, which is not 50 years, okay? So here's how we did the math magic. And a lot of this history actually came from Art Humphrey, who had many records about the course. And so according to Art, the course was first started in 1962. We've already heard that Dick Matterlees was the head of the course. He was a professor of nutrition and food science at the time. Now, Danny actually kept records of every time a new instructor was added to the class. And according to Danny's records, the first 4 original instructors, besides Dick, were Art Humphrey, Cecil Dunn, who we heard about yesterday, and Gerald Hispaka from Wyeth Lab. And I wanted to put this there because from the very beginning, there was a characteristic of this course that has persisted to this day. That is that we bring in people outside of MIT to be involved, and we always have a connection between industry and academia. That was from 1962, and it continues to be the case, as I said, today. We had this picture taken at a special dinner that we had celebrating the 50th anniversary, 3 generations of Art Humphrey, who begat Danny, who begat Charlie, right? All together at one time, and really was a wonderful opportunity, again, using this excuse, as I'll call it, as the 50th anniversary, to just focus on a celebration of the legacy that Danny was leaving behind. In 1965 is when Danny actually joined the faculty of MIT and joined the fermentation technology course. And everyone agrees, there's no disagreement whatsoever, that he really transformed this and made it into a household name, as it were, amongst the biotech industry. He actually did not stay in the US. The course was offered in Europe and ultimately in 7 different countries. It was offered in Asia, multiple countries there, and it was offered in tailor-made format at several industrial sites too. So again, the point being not to keep the knowledge to oneself, but to spread it as widely as possible to those who might benefit. In 1978 was when the textbook that we've heard about arrived. And I just wanted to point out, here are the authors of the textbook. Here are the years that all of these authors actually taught in the summer course. Charlie is the reigning champion at 51 years and counting. We did not have the course in 2000, right? So this is actually Charlie's 50th anniversary of teaching in the course. And then you can see Art Humphrey, Danny's records on this were actually incomplete, and when everyone else was involved as well. Now, this was a little fun fact, which was that Art was positive that the course was not, I think maybe Danny's note said the course was not offered in 64 or 66. That's how we got from 62 to 2014 being 50 years, because if you take out those 2 years missing in the middle, you get 50 and not 52. However, the year after the celebration, someone sent to me their course certificate from 1964. So I still have no idea what we were actually celebrating in 2014, but I do know we had a marvelous time, and I have the pictures to prove it that I'll continue to show you in just a minute. Henry Brown, by the way, is a 1956 practice school alum of our department, and he found that as he was going through, I knew him through some other connections, alumni connections, and he found that as he was going through some old papers and took a picture and sent it to me. The course up until 2014 had 28 different instructors who had participated, and I've shown here those instructors who were teaching in 2014, including Charlie, Jim Leong, who many of you know, Bob Kiss, who's come up many times before, and there's Morris Rosenberg, and then again there's Art Humphrey. Now, I have since then included additional instructors, so we now are at 30 different instructors having participated. At MIT, more than 3,500 participants have come through with a maximum class size of 155. There's been estimates of about 300 people participating at industrial sites, another close to 1,000 in Europe, about 500 participants in Asia, which really means this is a course that has more than 5,000 alumni and continues to increase its numbers. Again, emphasizing that these fundamentals are very important towards nurturing this industry that Danny helped to create. We did not count the lobsters, but as someone pointed out yesterday, there were always lobsters, and the lobsters were always greatly enjoyed. And we did really use this as an opportunity to celebrate. So we had a big cake. We actually gave out mementos, bottles of red wine that were engraved to celebrate the 50th offering. We didn't call it a 50th anniversary. It was a 50th offering to try to make the numbers work of the summer course. And I want to say, in closing my remarks about the course, that it really has been a labor of love for many different years, and it has taken a village, or I say at least a chief wrangler. Here's Susan Lanza, who many of you know. Her daughter Amanda is in the audience today. It's her husband Robert and her other daughter Jennifer. Susan was really the driving force behind keeping things going for so many years. And once Susan retired, Gwen Wilcox, who many of you have met outside, has been helping me along as well. And I love this picture because this is Rosangela DeSantos, who, again, many of you may know, worked with Charlie for many years on the downstream processing course, which has always been a companion to this course, and we always treat them like this. It has also been a family affair. From the very beginning, a family affair, and I'll say more about that in a little bit, but here's Vicki with Danny, Sheila Humphrey with Art, and there's Peggy, who many of you saw last night with Charlie. Now, the last thing I will say before I wrap up my remarks with a few more personal things is that I did have the honor of co-advising Danny's last graduate student, Andres. We published a couple of papers together with co-authors, and I sat in many meetings with Danny and Andres. Jim Leung joined us for many of those meetings as well, and Jim and I continued to advise Andres when Danny was physically unable to. And we had, again, at our dinner table conversation yesterday, lots of discussions about what the old days were like. And I will say only one time that I have to say, Danny, you can no longer say that to a student. Right? So times did change, right? He was a little bit softer. He was still a little bit on the gruff side, but times definitely did change. So this is my favorite picture from that night 7 years ago. I don't know what Danny said to make both of us laugh, but I do know it was Danny who said something that made both of us laugh. And I really appreciate the joy that I see on his face in this picture. Because he really loved this course. He really loved teaching. He really loved educating and reaching out to share what he knew. And so I want to just talk for a moment about the things that I learned from this. One thing I learned was that family was first. So I mentioned this previously, but every year when we were doing RSVPs for the course dinner, the question was not just, is Vicki coming? We knew Vicki was coming, but is Keith coming? Are the girls coming? Is the family coming? And it was always about a family affair. And because of that, my own kids have grown up going to lobster dinners, and Danny loved seeing the girls. They're not so young anymore, they're teenagers, but he asked about them constantly. And you heard about this from Jono yesterday, that he really was genuinely concerned about the challenges of being a working mother under the stress of the tenure system in academia, but also at MIT. And he was very much a supporter and a champion to do whatever he could in order to make life easier for me, and as you heard yesterday, for lots of other people. He also showed me that you get to choose some of your family. I mentioned when I first came back for a visit in 1998, Danny introduced me as his advisee, and I said, no, you weren't my advisee, I was in your class. And he continued throughout his days to refer to me as his former advisee, and so I decided, okay, I'll take it, right? I prefer to think of myself more as a daughter, but it let me know you do get to choose these relationships, and these academic relationships you have can be very nurturing and very fulfilling and sustaining for a long period of time. And so I've always been proud to sort of say, well, he wasn't really my advisor, but he kind of was my advisor, and he's always allowed me, or he's always referred to me in that particular way. He was tough, and he was critical, but he loved a good sparring partner. So you heard yesterday about how he would seemingly be sound asleep at seminars and then wake up and ask a probing question. This happened to me during my faculty interview where I was talking about these things I was making in E. coli cells, and he woke up and started talking about back of the envelope calculations and went on and on and on about how what I was talking about was just physically impossible and blah, 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 blah. And I still remember I said to him, it sounds like perhaps you have answered your own question, but if you would like an answer, I'm happy to give you one. And very quickly, I'm talking about polyphosphate and not phosphate. And he said, oh. And I think that's the moment our relationship changed. Because from that time, he knew he could say whatever he wanted, that if I could defend myself, I would defend myself, right? And if I couldn't, I would admit to it, right? He was one of my mentors, as Paula mentioned. And I remember one meeting in particular where I walked in and said, okay, I know I need more papers. Let's talk about things I don't know. And I think that's when I got a little bit of additional respect as well. The last little story I will tell goes back to what I said before, which is that Danny told me this microbial stuff was going nowhere, and I needed to learn mammalian cell technology. And so one year in the summer course, around 2010, Danny came in to give his lectures, and he starts the lecture by asking, how many of you are working on mammalian cell systems? And he had them raise their hands. And then how many of you are working on microbial systems? And he had them raise their hands. Now, he did not know I had asked the same question the day before, and I knew the answer. And so when he has the hands, and it turns out 70% of the class is working on microbial systems, he looks at me in the back of the room and says, I guess you won. And then he started delivering his lecture. So I've talked about these different flames, and I think those have come together in this, if I go back a second, in this passing of the torch. I do feel like Danny has entrusted me with a big part of his teaching legacy. I said before, a big thing I learned is we are good teachers because we want to teach, not because we have to teach, and I have very much tried to instill that in everything that I do. And the last thing I want to say in closing is that this slide here, I increment the offering, but this is from 2021. I show this slide at the beginning of every fermentation technology class, where I refer to this not as fermentation technology, but as Danny Wang's fermentation technology, because that's the first way I ever heard it phrased. When I worked at Merck, and Merck was sending scientists to MIT for the summer course, it wasn't, are you going to the fermentation technology course? It was, are you going to Danny Wang's fermentation technology course? It turns out I am forbidden from changing the name to Danny Wang's fermentation technology. I checked and was told no, but I want to make sure that everyone here knows that Danny's legacy will not be forgotten in this course, and it will forever, for us here at MIT, be known as Danny Wang's fermentation technology course. Thank you very much for allowing me to share my remembrances. And I think for the sake of time, since we started a little bit late, we'll go straight into Nubar, and Charlie Nubar and I can do questions together at the end. Thank you.
All right, everyone. It's been a very exciting morning so far. And now to close out our discussions this morning, I'd like to introduce Nubar Fayan. Nubar did his undergrad work at McGill University before coming to MIT for his PhD in chemical engineering, where he was supervised by Danny Wang. He dedicated his career to improving the human condition by systematically creating science-based information that serves as the foundation for startup companies through flagship pioneering, where he's helped to start more than 50 public and private healthcare and life sciences companies. Nubar also serves on the boards of a number of public and private companies, including Moderna, where he is chairman, Rubius Therapeutics, Omega Therapeutics, Tessera, and Seventh Sense Biosciences, among many others. Nubar is also an active member of our community here at MIT, including a member of our visiting committee, and in 2016, he was appointed to the MIT Corporation. He is a passionate advocate of contributions of immigrants to economic and scientific progress. Nubar received the Golden Door Award in 2017 from the International Institute of New England in honor of his outstanding contributions to American society as a U.S. citizen of foreign birth. He was also awarded a Great Immigrant Honor from the Carnegie Corporation in 2016, and he received a Technology Pioneer Award from the World Economic Forum in 2012, and was presented with the Ellis Island Medal of Honor in 2008. Please join me in welcoming Nu Barafan.
Well, thanks, Paula, and it's been great listening to everybody's experiences and kind of sharing this moment. We've all looked forward to it, and the pandemic delayed it by 2 years, just like that 50th celebration. But here we are, and it's great that we get a chance to do it. You may have noticed when I walked up, I put my bag there. I think that's about where Danny would have sat in a BEAM seminar. And so my intention is to largely give this talk as though he was in the room, because I think if I could give a talk that would be interesting to him, it may also be interesting to you. Unfortunately, in that regard, if he was sitting there, I don't think he would have allowed many of you yesterday, or even Kristalla now, to talk so much about him. So I will actually limit my comments that are aimed at him and talk instead a little bit about things that might have interested him. All right, so you've seen this kind of the elements of this slide before, but I just wanted to summarize that in doing that tribute to him and looking at every single paper he had authored by the time we published this and making some of these charts. What I realized in the top part is that he was curious, he had attention deficit, and he worked on a lot of different things. And the irony of it, and I've reminded him many a time, is that the first few times I met him, after 2 or 3 times when I started working with him, he told me that I was like a dog, a flea on dogs, going from one dog to the other, because I kept changing my idea of what I want to work on. And then when I realized what he had done for most of his career, going from one thing to the other, I realized that it was the pot calling the kettle black. But I'm very proud of that, because what you'll see me talk about now will completely suffer from the syndrome of being a flea jumping in dogs. But he was that. He enjoyed the diversity of experimentation and thought. He also kind of obviously stood for excellence, but also consistency, which is something we all experienced. The time he came to work, the things he would say, just very, very repeatable kind of. But at the same time, what I didn't realize is this was the fun part of plotting, making this plot on the bottom, is that he, with a 0.98 regression coefficient, published 6.37 papers per year throughout his career. It's quite remarkable, actually. I don't know that he thought of it that way, but there it was. And then you'll see that period after 83 with the acceleration due to my colleagues who joined around that time. And then, but it's a very interesting thing about him, and I think that is reflective of his personality, of his work ethic as well. You also saw this tree, and I think all of us, when we see this put up, try to figure out where we are, and you realize how insignificant you are, but then again, you realize how significant you are, because every single one of these fruits on this tree, and then their descendants, are actually trying to propel his legacy, as we've heard about before. I actually found two photographs which you haven't seen probably for a very long time that were significant to me. For some odd reason, the 50th birthday, and Greg, I have a picture of you as well in this same celebration. I think you were sitting up there. Was the only time in my life where I actually had a kind of a beard. It's not really a real beard. Danny hated that, but that's why I kept it until I was through the 50th celebration. And so there's a picture, and he was in his glory as we celebrated him. And then this is a picture very soon after my PhD thesis, and just as I was starting a company, and you'll see Danny and you'll see Keith, who actually worked in the company for a summer, actually painting walls and helping fix things that we needed fixed, etc. And it was great to have Keith kind of grow up in the same environment, and those are my parents. All right, so from there, I actually want to jump all the way to the present, and in thinking about what to talk about, future-looking kind of. And also looking at the present, I thought I'd say a few words about the experience with this technology that led to the COVID-19 vaccine, or at least one of them. And I thought I'd put a little bit of context and then focus on a little bit about innovation, the way the team that I work with has been practicing it, and then think about where things may go in the future. But one of the things that I thought would then be useful is to just kind of contextualize an event that happened less than a year ago, which was the approval of a vaccine that had not been thought about more than 9 months earlier, let alone worked on for much more than 6 months in any clinical work. And this connects to Danny in an interesting way, because 10 years earlier, in 2010, when we started what became Moderna, one of the first people I actually talked to was Danny. And I kind of went to him with great trepidation, because I used to tell him all the different things we do in their nascent stage, just because he was interested scientifically. And this one was a little bit troubling, because the premise was nothing short of basically obviating the need for fermentation and purification in the way we knew it. You know, I spent all of my life working on making proteins and purifying proteins and analyzing proteins. And while this would involve a little bit of analyzing proteins, it would do away with that. The reactor would be the patient, the subject. And I kind of thought he's going to, I thought I was going to meet the same fate as microbes, so don't work on them. But actually, he was quite intrigued. He said, no, it would be interesting. He said, that's what people said about DNA in the beginning, that we're just going to stick DNA in. Who knows? But he was actually quite intrigued at the prospect that the large amount of innovation that he had overseen and contributed to would suddenly be outsourced to the body, which actually knows how to do all these things with great precision, greater precision and predictability than anything man has made outside the body, but with other sets of technological challenges of how do you actually design, make, optimize RNA, let alone deliver it to the right cell safely, et cetera. So that's the journey that took 10 years to get to this point. Just to put that one date, December 18th, for us in context, the journey started back in January 11th, when the sequence of the virus was revealed online. 2 days later, we had the design of what is actually in the vaccine today. In other words, we went into labs and made the design. We never really changed the design. We didn't change anything. There was no optimization. We had no time to optimize, and I'm not sure we had the need to either. But certainly, if you have a platform where you've done thousands of sequences before, it's a little different than if it's a bespoke thing, the way biotechnology had started 40 years earlier, where every single protein you started from scratch. Here, you were just moving bases around, but it was really highly precedented, at least in our labs. And within a couple of months, we had entered human trials with man. So March 16 was the first person who received the vaccine for a virus, the sequence of which was revealed 44 days earlier. There was a lot of collaboration. This chart is actually showing the virtues of the conditions that the pandemic forced on us and the degree of capricious lack of collaboration that pervades this space that went out the window. When you're huddling at home with your family and you're afraid to go out to get food, turns out you collaborate. And so large agencies, government, regulatory agency, NIH, clinics all around the country just gloriously came together and said, let's just do whatever it takes. We've got to do this without any delay. And that they did. Without all of that, without any of that, none of this would have worked. And so you'll see collaborations with government, with BARDA, with the COVE study that the NIH undertook, with Lonza, the contract manufacturing group that really was a great collaborator, because they were the recipient of the only technology transfer we ever made, because we needed competent counterparty to actually learn how to do these things. It was the first time it was being done beyond our walls, and so forth. And then it continued. There was further work done with boosters and the like. And I think since this has been well covered, I won't bother you with it, but it'll continue. So this is, we've done quite a bit of work on variants now and variant vaccines as well, just in case the virus doesn't actually want to follow our timetable of retreating when we're sick and tired of it. There is a kind of an interesting narrative emerging that the viruses must be reading social media and saying enough already. Now this is it, that's it, no more waves. If you wake up this morning, Germany had a single biggest case number in the history of this pandemic yesterday. And so we may or may not get through this without a lot more turbulence, but clearly we can't afford to say, stop working on variants, stop working on other vaccines, because this one we're done with. We have a vaccine that we've tested in humans for the beta and the delta and the combination in case it's ever needed. Of course, there's a huge overlay in all this, and I wish I had Danny to talk to about this. Well, he's kind of here, I guess, in memory. about the degree of which politics and society and human beings get involved in this, in the science, in the technology, in decision-making in times like this. That we weren't that prepared for. I don't think I took a whole lot of humanities courses or political science courses, but I can tell you the last 18 months has been the merger, not just the convergence Phil talked about between science and technology and patients and doctors, but also of regulators and governments and the media that amplifies truth, the media that amplifies mistruths. It's just been a remarkable thing. And I'd say, to give a lot of credit to the fact that I never really moved more than a mile away from MIT for the last 38 years, a lot of what MIT had taught us, but also continues to teach us, if you continue to affiliate with it, is that. Because there are people on campus who are dealing with every single one of these issues. So it's been a very rich place to rely on for thought about how to navigate this. Now, just kind of in a very simplistic level, I always want to put up kind of what's all this about, right? We grew up kind of convincing unsuspecting mammalian cells or bacteria to make proteins that we wanted as best we could and as pure as we could. And it was basically enabled by what we learned about DNA and its kind of transfer of information all the way to proteins. But this middle molecule, mRNA, thanks a lot to Phil's work and others, which we had understood, described, and a lot was known about this magic that it does, sitting in between the information molecules of storage and the effect that they produce through proteins, had not been used as a therapeutic agent for all sorts of reasons when we started. And what we set out to do was, in fact, figure out how to tame, if you will, this molecule in a way that you could deliver it to human cells, preferably the cells you want, to the extent you want, in a way that does not cause the immune response to think that it's a virus, which is what it's prone to do, because there's lots of RNA-based viruses, and therefore lots of innate immune responses intracellularly that will respond to it as though it's a virus, including interferons and everything else that were well understood. But the mechanisms for just single-stranded RNA were not well understood 10 years ago. And to be able to code then through software, if you will, to make any protein you want, whether it's secreted, cell surface, mitochondrial, nuclear, etc. And that was what we set out to do in the beginning. That was not something that we eventually thought through. The goal was to say, could you kind of create a ubiquitous portal of information into cells? If so, which cells? Would it be muscle? Would it be liver? Would it be, circulating cells of the immune system, etc.? So that was kind of what we set out to do. And by and large, over a 10-year period, that's what the company had been well on its way to do. The project started in our labs and flagship in the summer of 2010 in a project called NICO LS18. As you'll see a little later, we tend to number our projects. This was the 18th platform project that we worked on. In fact, John, I don't know, John, if he's in the room, I saw him outside, was actually there he is, was in the process of leaving Merck at the time, and I quickly kind of convinced him to come and help, given that nobody, I remember him saying, well, he hadn't worked on RNA, he'd worked on DNA before, and I said, yeah, but nobody else has either. We're going to have to figure this out. So that kind of brave new frontier, which is what we saw at MIT with mammalian cells to begin with. He's one of the first graduate cohort who worked on this. It was the same with RNA. We just had to figure out how do you make it, how do you stabilize it, et cetera, protect it. The very first patents that are the basis for the platform, the company, were filed in 2010. They're in fact not the patents that preceded, because there's lots of IP that has been talked about in the past. But what we needed to do is to enable the use of these as drugs, not the use of these as cell-based manipulation reagents, which had been done and shown before. And what followed were also collaborations. You'll see that AstraZeneca took a major leap of faith in 2013 in partnering with a company that had shown some mouse data and a little bit of non-human primate data. They deployed $400 million over a period of 2 years to enable our platform, and in return get options to some of our programs. Merck followed suit with vaccines. Vertex in rare diseases, those continue. Vertex in particular is continuing to grow. The very first, interestingly, you might find, I don't know if this, yeah, there we go. The very first use of mRNA in humans that I know of, this form of modified mRNA with LNP, was actually, ironically, in Germany by us for 2 strains of influenza that have not entered humans before. These were potential threatening pandemic or epidemic strains. And the reason we picked those, we thought it was the safest thing to test this on. You have naive human immune systems that we could give slight variants of the antigens to, and then you see their immune response, and you can then test whether you can do a neutralization. You see whether the antibodies would be neutralizing. And that was, we were testing for safety and neutralizing antibody. And we saw them very robust antibodies. In fact, from here to 2021, what improved was the LNP technology we used, but not much else. We already knew in 2015 we could do this at least in 2 influenza strains. What ensued was 9 additional vaccine programs. I don't have them all listed here. And in fact, started doing work in cancer and cardiovascular disease. And by the time COVID came along, this is a little known fact, the company had already done 9 different vaccine human trials in phase 1. Every single one of them made neutralizing antibodies at the level that had been previously shown to be sufficient, relevant for that particular virus. And so even though the pandemic was a big kind of a scary proposition to go after, in fact, we weren't starting from scratch at all. There had been $2 billion building this platform spent, and there had been 16 development programs already in the clinic, most of which were in the vaccine area. I just thought it would be interesting to know the background. So where are we today? That's another thing. Everybody says, well, now that you've done this, what else are you going to do? Well, we were doing already, by the time last year ended, 30 programs, clinical programs, the majority of them, more than 50% of them, already through phase 1. And this is how it's kind of broken down. We have a number of prophylactic vaccines. That number is growing considerably. We have a number of things that are being made, IL-2, for example, in autoimmune diseases in clinical trials. We've been working for years on cancer vaccines, personalized, tumor-derived, 35 to 50 different antigens in the form of mRNA in a vaccine given back to patients to see whether we could get the immune system to go find some of those neoantigens expressed in the patient's tumor, and that's continuing. I'll say parenthetically, by the way, those of you who, and again, John will know this, those of you who kind of would think about how did this process development happen so quickly, turns out that making the personalized neoantigen-based vaccines that we've been working on allowed us to respond very quickly to this virus. We just thought the virus is yet another neoantigen, meaning we had the workflow to go from sequence to design to getting it into a vial. In this case, it was a couple of hundred vials for one patient, let alone for a couple of hundred to do a phase 1 trial in. So the actual workflow we had working in Norwood and the process was the identical process that we used in the first wave of phase 1 materials because of the work we had done in cancer vaccines, which is kind of very interesting kind of translation across applications. We've been working intratumoral. The delivery is always a very interesting challenge here. So intratumoral targets lend themselves to this kind of approach easier than delivery might. Just not to go through every one of these, quite a diversity of applications, and we'll see. Over time, a subset of these will actually gain clinical traction, and you expect will become a whole new class of medicine. So, question we get asked a lot is, how could we go so fast? I'm not going to spend a ton of time, so I'll go even faster through this. A lot of things. A lot of things. And what's not here is actually kind of that Murphy's Law seemed to be on sabbatical. Because a whole lot of things had to go right for this to work, and actually all of them did, and there's really no good explanation for that. You could say preparation, hard work, and I agree with all that, but in the control experiment where we also are highly prepared and do hard work, it often doesn't really work out. So the speed here was a function of the fact that, say, first and foremost, that this is innately an information molecule. This is not an analog situation where every protein is kind of interesting drawing-wise to another protein, but it's got nothing to do with another protein. Here, the mRNA molecules are kind of well-behaved among each other as it relates to their use, particularly for vaccines. That's at least was our experience before. We had worked actually on MERS with the very team, Dr. Fauci's team, that was at the command center of all this. And in fact, the very same people we'd worked with on a pre-fusion spike protein vaccine, on which we have actually Moderna filed patents many years ago. There's an approved patent on a pre-fusion protein as an antigen together with LNP from years ago work that we had done on MERS. And in fact, this is a very related application. So we had a big advantage in the work we had done with them in terms of the biology and also the clinical, kind of getting ready to do clinical testing. Having our own manufacturing facility helped us a lot. I think that if we actually tried to start from scratch. We might have been lucky to find a pharmaceutical company that was doing it. There weren't a lot of pharmaceutical companies either interested in or getting ready to work on mRNA. It was largely discounted as a new modality, just like siRNA had before and antisense had before, and antibody-drug conjugates had before. I mean, our whole history has been one of large pharma companies waiting a long, long time to actually think something's going to be commercially viable, and in fact, they've been mostly right, unfortunately, for those of us who are engineers in the room. In this one case, though, that made them have to sit on the sidelines, and with the exception of Pfizer, who made a very good strategic deal, really most all of them were not able to enter into this space. And then the last thing I'll say, and I won't get into this much, but I can in the Q&A, is the company, thanks to the leadership of Stéphane Bancel, who joined very early on, was built as a digital company. And I've never really seen in the other 100 or so companies that Flagship's been involved with, most of which we founded, that level of native digital implementation from day one. And it's really remarkable to see what that means. In other words, if you say that everything has to be done digitally, then the quality control people are actually using algorithm development to be able to improve the way they're doing things, the connectivity, the Internet of Things that you put into every single aspect of what you do, so you have the measurements, whether you know what to do with them or not. All of that was in place. Don't ask me why, because it looked like overkill and it looked really expensive and it's not something that small companies do. But boy, did it come in handy to allow us to go faster, because we did not spend a lot of time gathering disparate data and figuring out. It was just all already, the substrate was completely there. And I will mention one interesting thing, particularly if Wei Shou is listening in at this point, which is that there is an interesting connection between Danny and Moderna, not just through me, not just through Bob Langer, who was an academic co-founder, but also through Stéphane Bancel, because Stéphane Bancel did his master's thesis with Wei Shou Hu in Minnesota. And when I first met him, that's actually was our connection, because I realized that he came from a good pedigree. And so there it is. And he's a biochemical engineer, by the way. He has the same training as we do. So let me just kind of accelerate and tell you a few additional things. I've got some more time. Just for variety now more. I think that all of this is put us into a new era of where immune medicines will actually gain a very different level of importance than just an antibody for a target. Because actually, I would contend to you that our immune system is a more potent pharmaceutical technology than anything any human has created. We just haven't known how to deploy it in a controllable way, in a predictable way, as broadly as we've shown the ability to at least do in one case very quickly. And so the question is, what else can our immune system do to protect us, or to delay the progression of disease, or to fight or cure a disease? I think in that book, what we've done in the last 20, 30 years will be kind of the first page of the first chapter. And I think if a Danny was starting out today, and there are many Dannys starting out today, I would say that the application of biological and biochemical engineering to enable immune medicines, this is not just my idea, but it is definitely in front of us, because it requires a knowledge, a model, the way the BE folks talk about measure, model, and manipulate, that is needed in order to work with these kinds of medicines. It's just not a cause and effect. It's not a molecule that binds another molecule and you hope to God everything else kind of is a side effect you can manage. You're going to have to actually use the system integration that the immune system uses in a safe way. And so I think this is a very exciting period that will be enabled by code molecules like mRNA and others in order to go after not just infectious disease, but I think many other kinds of diseases. For example, we're working on whether there are safe ways to have a controlled, non-autoimmune-like, but immune response to proteins that we want to lower. Can we get our own immune system to lower our level of PCSK9? Can we get our own immune system to lower our level of interferon or of TNF, if that's what we want to do? It seems dangerous, but not if we can actually control the variables and go in and do this. The immune system can do that. It's just that if you don't control it, it's going to go after it in many other ways too. So more to come in the future, but very exciting. I'll say a little bit just about how Moderna, if I zoom out, like I said, is number 18 of a large number of these things that we've done both before and since. And a big part of what we've practiced, and again, this stuff now being a little bit more has been written about this, so if anybody's interested, I'm happy to refer you to. But one of them is about, and we heard this about Danny's kind of encouragement we all received, which is that you've got to be able to make leaps. And if all you're working on are things that are kind of obviously the next adjacent thing, then you're not going to be able to kind of advance things. And so you've got to, once in a while, be able to go after the tough problems we heard about and the important problems. Well, the important problems usually are not sitting there really adjacent to what you already know how to do, right? So going from making proteins to mRNA, that's not an adjacent problem. That's a pretty big leap. And if you say, well, no, I'd rather work on a protein with another thing stuck to it because I already know how to make proteins, that mindset, one of the things we've realized over the years is that innovation, which tends to happen from the core, that is whatever is known today, outwards through adjacencies and through making bets on whatever seems to be both diverse enough but also proximal enough so as to be reasonable. So I will call this the zone of reasonableness. When you go to experts, experts can reason on a zone of reasonableness. Beyond that, they think it's unreasonable because their expertise does not allow them to give you a good estimate of risk and reward. But we know that over time, most of the exciting things will be outside. Over time, has to be. Because what's the chance that the only exciting things to work on are really, really proximal? Well, the question is, how can you go out to beyond these adjacencies? So I think that's one area that we've worked quite a bit on thinking about. And this is kind of a cartoon I've used for at least 20 years, and I keep coming back to it. When you get into the leaping business, that is, if you're willing to make these big scientific leaps and technological leaps, or at least try, everything looks like this kind of cartoon which says this could be the discovery of the century, depending, of course, of how far down it goes. And that's the reality. If you need to believe that this is a major new archaeological dig before you dig further, then you will constantly talk yourself out of it because you'll say, yeah, but what's the chance this is the big one? And so you'll just move on and do work on things that are more known to be big. And that means proximal, that means commodity, because everybody else is working on the same things. So we have a process that I'll spare you just because of the time today, but suffice it to say that it's heavily, which is ironic given my engineering background, based on evolution, actually, as opposed to engineering. So I've come to view Darwinian evolution as a powerful, powerful search algorithm of thought, not just of DNA. And so we use this at a very large scale. So for the last 20 years that Flagships existed, everything we do is predicated on making some general leaps through hypotheses as to what might be possible without any bound to what is reasonable and what is already precedented. And then we protect ourselves once we try to land through variation and selection until things emerge that are worth reducing to practice, further converting to platforms, and then eventually to products. So this is the process we use. And the other thing I'll say about Flagship, just, again, I'm saying this to Danny, so he already knew some of this, but this is an update for Danny, so you're just listening in. The other thing that we've learned is that startups that actually drive a lot of the big leaps and breakthroughs in our fields, in many fields, actually have their own structural challenges, even though they're glorious when they work. And a big one of them is that they have to combine often disparate academic science and scientists, disparate entrepreneurial management. I mean, you saw Phil's diagram from Geneva 40 years ago. Keeping those group of people together and getting them to do things and agreeing, let alone doubting the venture capitalists, which you also hear Phil sharp about. I mean, those are real impediments to success because as soon as you hit a bump in the road, nobody believes anybody, nobody trusts anybody. And imagine that now at scale. It used to be it was done 5 times a year. It's now done 5 times a minute in terms of bringing these things together and starting companies. And so because of this disparateness of the interest and the source of the capital and the management and the science, we kind of observed that there may be an alternative way, one in which you kind of have all that under one roof. So what if you could actually invent, convert into a corporate structure, and capitalize, in the first instance, all 3 elements of what it takes to succeed, and in fact, in a startup. And in fact, that's kind of what we're practicing, have been for a while, particularly for the last 10 years. On the one hand, for example, we filed 200-300 patents a year out of our own labs. These patents basically have nothing to do with what was done the last year. In other words, it's not the same area that we're furthering. These are unrelated new explorations that are leading to patent filings. There's about 55 clinical trials going on in companies that have been conceived through these processes currently, and some 150 preclinical programs. On the other hand, We have a large team of people. We're about 250 people. About half of them are involved in standing up new companies professionally, regularly, and they keep learning how to do it better and better. And on the other hand, the very first capital you need to take these significant leaps of faith, in fact, I should go back if this works and just show you the one thing yeah, here we go. I should have just pointed this out. Imagine a talk, Danny would have been really upset at me for putting this here. I put this here just before the talk. I snuck it in because I thought he might not pay attention because he was sleeping. But notice what I've said is that everything we do starts with a leap of faith. If I can kind of impress one thing on you, and again, you don't need me to do any of that, but just I'll remind you so that you can remind, many of you have kids that are getting to an age where they're doing graduate work and they're coming out. I would say that we as engineers, as scientists, have largely lost the confidence to use our imagination. Because the fields that we're in frown upon people saying things that require imagination to either say or understand, because it's unserious. You're supposed to stick to the facts. You're supposed to say things that are connected to the facts. And if you said something, I imagine if there was a virus in our body that was good for us and not bad for us. Notice I just said something that many of you might say, what do you mean, the virus that's good for us? Viruses are generally, pathogenic and our immune system's fighting them. But are there commensal viruses in our body, right? And you would never find them if you assumed that there couldn't be because viruses are pathogenic. You would find them if you just allowed yourself to use your imagination for a little while. That's what I mean by leaps of faith. And I think if you're allowed to make some leaps of faith, along with rigorous experimentation, etc., the world is a little bit different. And that's kind of what we've tried to institutionalize. And to do that, you need to have some money. Because guess what? You cannot go to a grant agency or a venture capitalist or a large corporation and tell them to invest in your imagination. You're not an artist, right? They go, if I want art, I'll buy art from a painter and stuff, and maybe they're a poor, starving painter on the street, and I'll feel good about myself. That's not what I'm going to do with science. And yet, that's what you have to do with science to actually create completely unknown new value opportunities. And that's kind of what we've tried to systematize. As I said, the systematized part Danny would have liked. The rigor of how we do experiments, Danny did like. And the output, once in a while, is pretty interesting, and it ties back to the learnings that we got here. I'm not going to bore you with this. Suffice it to say that the kind of nightmare of an ecosystem that we run now is basically shown here. This is current. There are, at any given time, 10 or 20 things that are fighting for survival in terms of being a new leap, trying to prove itself experimentally in labs, 4 or 5 people. These are numbered entities. There are a whole bunch of things, maybe 20 of them now, that are wholly owned portions of our system, not yet spun out. This is about 1,000 people who work in this kind of a setting, about 250 within the actual science and company startup portion, and then the rest are in the actual projects. And then as things emerge, they become growth stage companies, and that's when they kind of start working within our ecosystem. A lot of interconnectivity. Interestingly, this is virtually what a pharma company might look like down the line, because we talk to the FDA every day. Just that we talk to them across 30 different entities, across 50 different programs. So unlike a given startup that talks to the FDA once every 9 months and doesn't know what the FDA thinks in between, we have the luxury of sharing regulatory information, manufacturing, contract services, finance, capital. One of these companies raises money, all the other ones are able to tap into that source and start knowing the folks. So it's been an interesting learning experience. Now in the remaining, let's see, few minutes that I have, and I really will do this quickly. Let me just tell you very, very quickly. So this will be a little bit like a museum walkthrough when you're running out of time and you just kind of look at the pictures and make what you make of it. I'll tell you some of the interesting cutting-edge things that we're working on. Again, unsatisfyingly quickly, but there's stuff you can read about these, so this is not a huge secret. So first, one of the things we're working on now in a project called Tessera is actually not doing gene editing the way this is shown here, the way CRISPR works, but actually doing gene writing. And the thing we started working on 3.5 years ago is the potential to deploy mobile genetic elements as a way to actually do specific gene insertions and writing and rewriting of DNA and RNA-based information. There's a very, very huge stretch of kind of biology and homologous mobile genetic elements that we can use with great homology, and they literally can insert very sequence specifically if you can program them to do that. Large chunks of information, not just a single base change, just a build slide which takes forever. One of the interesting things is that basically the abundance of genes in our genome, of the top 100 most abundant genes, mobile genetic elements are here. and yet we don't use those to do gene writing today. Though we're working on that in a pretty active way. Let me skip ahead. So this is, sorry, but I was going to spend more time on this, but I got carried away with the other stuff. This is always the problem with starting with Moderna, is that it takes up most of the time in the air. So one of the interesting things is we're well down a path of doing this. We've already screened some 20,000 such candidate mobile genetic elements to see what's the best way to do this type of gene writing. And so this is really in between editing and gene therapy. We want to be able to safely put more information in a place where the genome is accepting of it versus wherever it happens to go. That's kind of in a nutshell, what we're talking about. And so you can do this at the DNA level, it turns out, with lots of possible components available in nature, but you can also do this at the RNA level, and we're working quite actively on both of them. Second one I'll mention very quickly is an interesting take on RNA. It turns out that of all the RNA forms, which is kind of plotted as a percentage here in our body, some 2% become proteins. And the rest of them are kind of given names as glorious as long non-coding RNA, which is the vast majority of them. Imagine being called long non-coding, and let alone tRNA and siRNA and many other forms of RNA. And so we got interested in a form that is shown here, which is that there's a massive amount of circular long non-coding RNA. If you look at red cells or platelets, upon enucleation, these things don't go anywhere. They just sit there as you can't degrade them. And we got interested in them because we thought, well, geez, why are they not degrading, and yet mRNA degrades in 5-10 minutes in a cell? And so what you realize is that these things are highly stable, they just don't translate. And conversely, what you really want to do is to be able to translate, at least for one set of applications, but these things are very unstable. So we decided to engineer translatability into eRNA. What we call eRNA is endless RNA. And that had not happened in mammals before, but it has happened in plants, for example, and it has happened in other systems. So we basically engineered these things, and what we observe is that you can now design constructs. Let me just kind of jump through it because of time. That in an apples-to-apples comparison, I'm not showing you therapeutic things because I can't do that yet, but this is a kind of a simple example. This is luciferase. If you do this at the level of mRNA with modified mRNA, that's kind of meant to last a while, you can get some level of protein expression for a while. And if you can engineer an endless RNA and make it translatable now, which is again a function of translatability, what we saw early on was the expression for tens of days, and in fact now we have some that have expressed more than 100 days out of a single injection. And you might wonder, can you do this over and over again? And so it turns out that you can do this over and over again, and not only that, but 200 days later when you do this again, you still get another 50 days of expression in a single injection. It's just not feasible with, and by the way, mRNA is not made by our systems to last a long time because you always want to be making whatever you need so that you can control and repress and regulate. These things you're not going to want to regulate if it's just an addition of a protein that you want to effectuate. So the therapeutic use might do that. Third, and I've got one more and I'll stop. Third area to kind of explore, Danny, is kind of what it means to do real generation of biology. So we think about engineering of biology, but of course biology is largely a generative system in that it can create novelty. And you can call it engineering, but I like to think of it as a generation. And you may know that in the machine learning AI fields, in addition to the pattern recognition that these techniques are very, very good at, which is in and of itself a great use to do gene pattern matching and looking at what matters. The other thing they can do is they can actually extrapolate and create novelty. And so you're seeing some of that in the technologies that are being used in face generation. So as you may know, there are many, many computer-generated faces now that you could never tell is not a human being. And so a lot of the faces that you see in the social media don't exist as human beings, and you cannot tell them apart. So kind of if you want to do a test of human intelligence, the generative systems can completely fool humans as to what is and isn't a human face. And so the question we got interested in is, what if you could actually use generative algorithms to actually create de novo proteins for any desired function? The way we do that today is we find the protein of that function and we change it a little bit. But what if you just started de novo? Could you computer-aided design de novo a protein? And that is something that it turns out to be, obviously has been something we've wanted to do for a long, long time. We can do it with DNA. We can pretty much code DNA. But can you code a protein? And to do that, at least what you need to be able to do is to show a learning algorithm enough instances of the function, not maybe precisely, but at least generally, enough instances of what a human protein looks like, because it turns out that we may not know what makes a human protein human, but the DNA level information turns out can discern a human protein from a non-human protein, and we have tons of proof of that. So you can train on the DNA code of a human protein, and the next one you show, the system could tell you this is likely a human protein, this is likely not a human protein, it's a zebrafish protein, at the DNA level, not at the three-dimensional structure level. So that's an interesting capability, so we started playing with that, and also about 3-4 years ago. And what's being generated in this company called Generate Biomedicines is a very interesting set of capabilities. We are now generating de novo proteins that bind to targets at just the level of DNA, so we can write the DNA code and go test it, and it'll bind with kind of submicromolar affinities out of the gates, and if you do one generation later. This is not screening, this is not directed evolution, this is just the computer telling you this will bind to that. We use it a lot in the COVID instance. There's actually a paper that was published with the Gates Foundation, large consortium, where we generated a bunch of pure de novo proteins to go after the spike protein wherever we want it at different places. But also, we're also training it to be able to make antibody agonists and peptides that are, again, de novo generated. This is not, look at this peptide and make me another version of it. And so we think over time, whether it's a year, 2 years, 5 years, 10 years, it's a little hard to predict because it's all a function of how much data can you generate to train how many systems to make how many proteins to evidence it. But I think it's not going to be very long before you can basically do DNA function learned systems that can actually start making, by the way, we've done this with enzymes as well. So one of the interesting things we've done is we've shown many examples of an enzyme and then said, make a completely de novo example. And we're about 3% sequence homology, it has activity. It doesn't have the same activity, but then 2 generations later, it has much higher activity. That's going to be the future in this space. And Generate's an interesting project for us also because we had Frances Arnold get involved early on, and her lab wasn't at that time yet doing this kind of work. They're now doing the academic side of this work, and of course, David Baker and others are as well. And I'll mention, this is not using folding. This is not DeepMind folding AlphaFold algorithms. These are trained to actually do the translational prediction, not the folding prediction after which you can make a protein. Let's see, I'll skip ahead. I think I'm done. This is the last little bit. Just in passing to say there's a company down the street that also has figured out how to do engineering of red cells that starts with donor, universal donor, progenitor cells, hematopoietic stem cells that in culture are lentivirally transduced, converted to be able to make any 1, 2, 3 proteins that we want, and then actually matured into a full enucleated red cell and delivered into humans. We're in trials with 3 different products in this area, applications of which in kind of multi-signaling, this kind of, imagine red cells with multiple signaling molecules going after either activating or deactivating immune cells or cancers. This is kind of what we're targeting it for. A ways to go to prove that it can be done safely and with a dose effect, et cetera, but we're, at least the engineering of this is within hand, and we think the scale up of it is increasingly becoming possible. We've got a long way to go in cost, et cetera. This is not T cells, these are literally red cells. And of course, it's also interesting advantages in that it'll only go where circulation goes, and we think we can do this, of course, allogeneically because we're using universal donor blood cells. Let me skip ahead. I'll end with this, which was a chart that many others used, but let me just end it with this one saying, which is, if you think in terms of a year, plant a seed. If in terms of 10 years, plant trees. If in terms of 100 years, teach the people, which is a Confucius saying, I think an appropriate way to end in paying tribute to Danny. Thank you very much.
Wow, that was an amazing view of where we're going. In fact, this entire session was amazing, and I'd like to thank all of our outstanding speakers this morning. It's been an honor to hear from you and to hear your reflections about Danny and his role in helping to project us forward. Now, before we break for lunch, and I'm sorry, but I think to keep on time, we should break for lunch and use the lunch as an opportunity to grab speakers and ask them questions. I did want to share some news that we broke last night at a small dinner for the speakers and Danny's family. First, I want to acknowledge Nubar Fayan, Jono Goldstein, and Charlie Cooney for their leadership in establishing the Daniel I.C. Wang Professorship in Biological Engineering. This chair, currently held by our distinguished scholar Chris Voigt, pays a permanent tribute to Danny's legacy as a leader in the field and one of the fathers of biotechnology. And I think Angie is in the audience as well. Hey, Angie. And is equally excited. She joins me in sharing our thanks to them. On top of that, Victoria Wang, Danny's wife of 54 years, has made a generous gift to the Institute in Danny's memory. This gift provides additional support for the Wang Professorship. It also establishes the Daniel I.C. Wang Fellowship Fund and the Daniel I.C. Wang Faculty Research Innovation Fund. The Wang Fellowship will support a cohort of students in both chemical engineering and biological engineering working on research and biomanufacturing, with preference given to young women in this discipline. The Wang Faculty Research Innovation Fund will support mid-career faculty in both departments, providing them with seed grants to really take on those exciting and innovative projects that they think of in the bioengineering space. The idea here is to give our faculty the freedom to think expansively and to pursue high-risk, high-reward new research that has the potential to shape both the discipline and the future of their careers. I think with Nubar's talk about getting outside of that ring and into the areas where you can actually see the frontiers, this is really the idea. This is an incredible gift and a fitting tribute to Danny's legacy at MIT. Danny will always be remembered here as both an innovator and a mentor of students. Now, on top of that, I'd like to thank Nubar for endowing our Daniel I.C. Wang Frontiers in Biotechnology lecture and in creating a new endowed lectureship in honor of Charles Cooney. So with all of these wonderful gifts, I'd like to thank our donors, all in memory of Danny. Thank all of you.